Good afternoon, everyone. I would like to welcome you to the first webinar of the Neuroimaging webinar series, jointly organized by EATRIS and EATRIS. Today, we are very pleased to have three presenters coming from both organizations, speaking about neuroimaging and CEST MRI in particular. My name is Rosanne Vechter, and I'm Education Manager at EATRIS. I will be hosting this webinar today. Before I introduce our speakers, I want to make you aware that this webinar is being recorded, including the questions and answers at the end of this session. It will be made available on EATRIS and EATRIS website shortly after this session. We have reserved some time at the end of the session for questions. We invite you to write down your questions in the question box in the control panel, as you can see on this slide. We have three speakers today. The first speaker is Martin de Kort from EATRIS. Martin is Program Manager, Translational Medicine and Drug Development at EATRIS. And after obtaining his PhD in bioorganic chemistry at Leiden University, he worked at several different pharmaceutical companies, including Organon, Schering-Plough, and MSD in leading optimization. He joined EATRIS in 2012. After developing EATRIS and its platforms as it is today, his prime interest is to match industry clients with EATRIS infrastructure experts in the most efficient way to start new public-private collaborations. The second speaker is Laurent Duquesne. Laurent is Project Manager at EATRIS and French National Coordinator for EATRIS. She's engineer in bioinformatics and modeling by training and has extensive experience in managing projects that specialize in bioinformatics, bioimaging, and neuroscience. And last and main speaker for today is Dr. Julien Flament. He is permanent researcher at MIRCen, the Molecular Imaging Research Center near Paris, since April 2012, where he is in charge of nuclear magnetic resonance platform. His personal research is focused on development of CEST imaging at very high magnetic field. The main objectives of MIRCen laboratory are to develop animal models of neurodegenerative diseases and to evaluate new therapeutic approaches. In this context, MRS, MRI, and particularly CEST imaging are developed in order to use them as valuable tools to monitor neurodegenerative diseases. Dr. Julien Flament has dedicated his entire PhD thesis on CEST methods that are now routinely used at MIRCen to monitor disease progression. I will now give the floor to Martin de Kort, who will tell you a bit more about EATRIS.
Thank you very much, Rosanne, for this kind introduction. And I'm happy to tell a little bit more about EATRIS before we hand over to our expert colleagues in EATRIS in Paris, France. EATRIS is a biomedical research infrastructure. It's an ERIC consortium in translational medicine. And its vision is to enable Europe with an optimal translational ecosystem. We do that by executing our mission, which is to support researchers in developing their translational tools and interventions for better health outcomes and society. We are located in 13 countries, and we have over 90 academic and non-profit institutions in our network. The central coordination and support team in Amsterdam is available to assist with expertise. If you look at the composition of the network, we are composed, we always say, of bricks and brains. The bricks are composed of 5 technology divisions, of which imaging and tracing is the topic of today. Apart from that, we also have a biomarkers platform with a lot of omics technologies and 3 product-oriented platforms in vaccines, advanced therapies, and small molecule. And the brains on the left hand ensure that the projects are performed in a compliant manner, moving from bench to bedside with particular attention to quality, reproducibility, and regulatory compliance on national and European levels. Legal and operational support is provided where needed to guide the contracting. Finally, we also have key opinion leaders that can be accessed and recruited to provide disease expertise, in particular in oncology, neurology, and rare disease. Obviously, today the focus will be on neuroimaging. If we look at the specific imaging and tracing centers that we have in our network, it's depicted on the map here. We have around 38 centers that involved and preclinical and clinical imaging, divided over 8 countries. They are all integrated in a multidisciplinary clinical setting, providing the necessary translational imaging in nuclear medicine and radiology. And these sites not only have a track record in preclinical research, but also are experienced in translating imaging tools, tracer, MRI sequences, or other protocols into the clinic. And again, the central coordination and support team in Amsterdam can be contacted for more details and can be used in dialogue to take on your request to find the right matching partner. Finally, in more detail, this slide capabilities that are covered by these and these can be divided in 3 main areas. The first is PET imaging of targeted drugs, biologicals, including the so-called immunopET and the radio labeling of antibodies or antibody fragments, DNA aptamers or nanoformulations. The second focus will be on small molecule PET tracers and quantitative imaging, ranging from preclinical rodent studies to non-human primate models and whole body clinical imaging of humans in early stage studies, including multimodal approaches such as PET-CT, PET-MRI, and as today's topic, CEST imaging. And finally, a network of ultra-high field MRI centers is available for more sensitive and early detection of disease. And in particular, the Neurospin facilities in France are pushing the physical boundaries to develop the most cutting-edge infrastructure. I think overall this concludes the introductory overview of EATRIS, and I would like to hand over to Roseanne back to continue the session.
Thank you, Martin, for your nice and short presentation on EATRIS infrastructure. We will have some time reserved at the end of both presentations for your questions. I will now hand over the presentation to our next speaker from EATRIS, Laurent Duquesne.
Hello everyone, thank you Roseanne. Before giving the floor to Julien, I will briefly present EATRIS, the co-organizer of this webinar series. EATRIS is the French node of European infrastructure EATRIS and is the only node whose specialization is solely neuroscience. EATRIS is also a national research infrastructure dedicated to neuroscience and more precisely to neurodegenerative diseases that are Alzheimer's, Parkinson's, Huntington's diseases, and multiple sclerosis. This consortium brings together all the needed bricks to build a full translational neuroscience project and gives you an easy access to multidisciplinary expertise and high-level platforms to achieve your project in neuroscience from bench to bedside. As you can see on this slide, EATRIS brings together multidisciplinary partners offering a unique synergy to carry out translational projects in neuroscience for academic and clinical collaboration, as well as industrial partnership. EATRIS is composed of expert teams from 5 renowned hospitals and research centers, accessible through a one-stop shop. This means that you can access all EATRIS core facilities and expertise with a single contract, and your project is managed by a dedicated scientific project leader. I will not develop each partner's capabilities further in this webinar, but all this information and more can be found on our website. Here is a brief overview of the neuroimaging capabilities EATRIS offers to its users. As you can see, it brings you all the way from cellular imaging and histology to preclinical and clinical in vivo imaging with state-of-the-art equipment. Scientific expertise is always paired with technology, allowing the development of new methods and applications. Among this scientific expertise is, of course, CEST MRI, the subject of this webinar. Should you have any question about EATRIS, please feel free to contact me or our business development manager, Ali. We will be happy to answer them or to put you in contact with the experts you need to define your project. I will now give the floor to Julien, who will tell us all about translational metabolic imaging using CEST MRI in neurodegenerative diseases.
Thank you, Lauren, and hello, everybody. Hello to everyone attending to this webinar. First of all, I would like to say that I'm very glad to present this webinar, which is entitled Translational Metabolic Imaging using CEST MRI in Neurodegenerative Diseases. And for that, I would like to thank EATRIS and EATRIS infrastructure to give me this nice opportunity. Just before starting, I would like to say a few words about the lab where I'm working. So the lab is called MIRCen, and it's the Molecular Imaging Research Center of the CEA, which is located in Fontenay-aux-Roses, close to Paris. MIRCen is part of EATRIS network that Lauren presented just before. And main topics at MIRCen are to develop animal models of neurodegenerative disorders, to understand biological processes involved in the disease, and to evaluate new therapies. In our team, we use MRI, MRS, and CEST imaging to study the metabolism in healthy and pathological conditions, especially in the context of neurodegenerative disorders. So MRI is a valuable tool to assess in vivo processes. So MIRCen is equipped by 2 horizontal MRI Bruker scanners, a 7 Tesla dedicated to monkey imaging, and 11.7 dedicated to rodents imaging. During this webinar, I will present the CEST MRI imaging modality, but I will try to highlight advantages of this method to study neurodegenerative disorders. So these diseases are like Alzheimer's disease, Huntington's disease, Parkinson, and so on. And they are characterized by a progressive loss of structure or functions of one or several brain regions. Neurodegenerative diseases are usually incurable, and some irreversible damages are caused to neuronal cells. And the consequences of these neurodegenerative processes are cognitive deficits, sensory and motor impairments, and alteration of behaviors. And their origin are very wide, like genetic problems, protein problems, or mitochondrial defects. The main goal of using imaging modality for clinical applications is to identify a relevant readout of the pathology. It's what we call a biomarker. And the ideal biomarker would be a molecule or an index measured into the image, and it would combine all these properties. So it should have a good specificity of the biological pathway, associated with low sensitivity detection to enable early detection. And the biomarker will also be predictive in order to optimize the diagnosis and to adapt therapy. It would be robust and non-invasive to ease its utilization. And finally, a good biomarker would be translational to ease the transfer from mouse or for animal to human, and to evaluate new therapies. Among all imaging modalities, MRI is one of the most promising techniques to monitor neurodegenerative diseases in patients. Indeed, with the increasing availability of high-field MRI scanners, now it's possible to acquire very high-resolution images and to monitor in vivo neurodegenerative processes. For instance, MRI is very useful in different neurodegenerative diseases like Alzheimer, Huntington, or Parkinson. So just to give you an example, in Alzheimer, it's useful to measure volume changes, especially into the hippocampus and temporoparietal cortex. And MRI can help to improve the diagnosis up to 87%. Also in Huntington, MRI is useful to measure changes of caudate and putamen. And there are also some modification of relaxation properties like T2 and T2 star signals. And finally, for Parkinson, we can also measure modification of T1 and T2 star signals, especially in the substantial nigra. And MRI is useful to measure targets for implantation of electrodes for deep brain stimulation. So, as I've shown just before, most of diagnoses are based on modification of structure morphology measured on anatomical images, as you can see here with an example in HD patient. Although very informative, such measurements are neither specific nor sensitive because they are only a reflex of long-term consequence of the pathology. In addition, they provide few information about biological pathway involved in the pathology. Another alternative is called MRS, for magnetic resonance spectroscopy. In this technique, we don't acquire image, but we acquire a spectrum that you can see here in bottom right on the slide. You can see different peaks corresponding to one specific molecule into the brain. Here it's an example of spectrum acquired in the mouse brain. And the advantage of this technique is that you can have access to biological information because you can measure, for example, metabolites related to neurons like NA or glutamates, or metabolites related to astrocytic compartment like inositol or glutamine. However, this technique suffers from a very poor spatial resolution and a priori positioning of the voxel. To understand the strong limitation related to the poor spatial resolution of MRS, we can consider, for example, MRS performed in a mouse brain. So, for example, if you want to assess metabolic alterations in the striatum that you can see here in green, it would be rather simple to place a spectroscopic voxel with enough volume, and here in this case, most of the voxel is filled with the striatum. However, if you want to do the same is a very heterogeneous structures like corpus callosum that you can see here in green, it would be much more complicated to place a spectroscopic voxel with enough volume. And in this case, only 15% of the voxel is filled with the corpus callosum. So we understand here that we need for a better spatial resolution. So to sum up about properties of the ideal biomarker, we would need a kind of more functional markers implicated in biological pathways and detectable with the good spatial resolution. And here comes the concept of metabolic imaging using CEST MRI. The acronym of CEST is for chemical exchange saturation transfer. To understand the principle of CEST contrast, let's consider two pools of protons. A pool corresponding to the free water, which are protons that we measure in MRI, and a pool of protons, which are in a different chemical environment because they are shifted by the CEST agents. And these two pools are in exchange. In a CEST experiment, we will saturate specifically the bound water of the bound protons that you can see here in red. And due to the exchange, the saturation will be transferred from one pool to another. But during the exchange, the saturation is still applied. So you're going to saturate again the protons. Now, if we generalize this principle to all protons, we see that a huge amount of the saturation will be transferred from the bound water to the free water. And the consequence is that the signal of the free water will be decreased. And this decrease represents the CEST contrast. To characterize the CEST contrast, we acquire what we call a Z-spectrum. It corresponds to the signal of the free water in function of the saturation frequency. And it looks like that. On a Z-spectrum, we see an absolute minimum centered at 0 ppm, which corresponds to the direct saturation of the free water, and a second minimum at delta B, which corresponds to the presence of the CEST agents. You can also see that the CEST contrast is hampered by the direct saturation. So to remove this non-specific contribution, we calculate the expected signal with no CEST agent, and we calculate the difference between these two curves. It's what we call NTR asim for asymmetrical Z spectrum. And this represents the specific contribution of our CEST agent. So thanks to the chemical exchange amplification, it is possible to detect very diluted molecules, which are classically undetectable by MRI. And this technique is also sensitive to physiological environment, like pH or temperature. The main advantage is the very good spatial resolution. And also, this technique is called on-off contrast, because you can only turn on or off the CEST contrast using the RF pulse. Many people wonder if CEST requires specific hardware or specific configuration of MRI scanner. And the answer is no. You can perform CEST imaging on all MRI scanners. The only thing is that you need a CEST sequence, but it's rather simple. You can select your favorite MRI sequence like gradient echo, turbospin echo, or EPI. And you just have to add, before the imaging module, a saturation module, which is characterized by only 3 parameters, which are the saturation time, the saturation intensity, and the saturation frequency. And by playing with these 3 parameters, you can target specifically different exchanging protons. So it means that with a very simple sequence, you can select different protons. I will explain you later how you can do that. And how you can use it on almost all scanners is highly valuable for translational studies, going from mouse to human. So CEST imaging is a very versatile imaging modality, as it's possible to detect several molecules which exhibit exchanging protons. For instance, it's possible to detect endogenous metabolites such as myoinositol, lactate, glucose, creatine, or glutamate. And interestingly, all labile protons exhibit different exchange properties like chemical shift and exchange rate. So it is possible to specifically image these metabolites by tuning the saturation module of the CEST sequence. So for this webinar, I will mostly focus on 2 metabolites, which are glucose and glutamate, because they are very interesting for neurodegenerative disorders. And first, let's see application of CEST imaging of glutamate. Among all metabolites, glutamate is particularly interesting because it is involved in several functions into the brain. For example, it plays a central role in brain energy metabolism, and it is implicated in neuron astrocytes interaction through glutamate-glutamine cycle. Interestingly, glutamate has been shown to be a good candidate for CEST imaging, and it has a lot of potential applications, especially in the context of neuroinflammation, HD or AD, and so on. And I will show you some examples later on. The first application is about the use of GluCEST to image astrocyte reactivity. So as you may know, most of brain pathologies, such as neurodegenerative disorders, induce inflammation of tissues. This process is called neuroinflammation. In response to pathological conditions, several brain cells become reactive, which is the case, for example, of astrocytes. You have an example here of reactive astrocytes on the right. And these cells are very abundant into the brain, around 90%, and they play a key role as trophic and metabolic supports for neurons. The idea here was, is it possible to use reactive astrocytes as a biomarker of neurodegenerative disorders? So for that, we use a rat model of astrocyte reactivity that which has been developed into the lab, and it is induced. The astrocyte reactivity is induced following injection of lentivirus encoding for a cytokine, the ciliary neurotrophic factor, or CNTF. And you can see on the right, the top right, using Vimentin staining, the zone of astrocyte reactivity. So first of all, we characterize the MRI signature of our model, and we show a significant increase of astrocytic metabolites like inositol and choline, and significant decrease of neuronal metabolites like Na and glutamate, and especially a strong decrease, 18% decrease of glutamates. So it means that this model is good one to evaluate CEST imaging. Here you can see 4 typical examples of Greasiest maps. We clearly see that Greasiest contrast is lower in the right hemisphere of all animals where CNTF was injected. Then we calculated the mean Greasiest contrast in a voxel comparable to the spectroscopic voxel, and we observe a significant 15% decrease in the CNTF side compared to the control side. And it was very consistent with the 18% decrease measured by MRS. So it means that we measure the same glutamate pool, but here we can really map the spatial extension of glutamate changes. Finally, after experiments, animals were sacrificed and their brain was extracted in order to perform Vimentin staining on each animal. We can see on the Vimentin the zone of astrocyte reactivity. And as you can see, with the dashed line, the zone of reactive astrocytes matched pretty well with the zone of lower Greasiest contrast. So here we show that it was particularly useful to combine MRS and CEST imaging to take advantage of the both techniques, so the quantification for MRS and the good spatial resolution for CEST imaging. But here we also show that Greasiest has a strong potential in order to image very accurately and to monitor the spatial extension of astrocyte reactivity. Then in a recent study, we also demonstrated that Greasiest could provide crucial information in the context of Huntington's disease. So just briefly to remind what is HD, Huntington's disease, this is a genetic disorder which is caused by abnormal expansion of CH triplets. So when patients have more than 35 repeats of the triplets, they will develop HD. And this disease is fatal 15 to 20 years after symptom onset. So this is caused by accumulation of mutant huntingtin, which is misfold, and which accumulates into cell bodies, and it leads to neuronal atrophy and then striatal atrophy that you can see here. So the idea here was, is Greasiest a good biomarker of HD? So to answer to this question, we use a mouse model of HD, which is knock-in 140 mouse model. So it's a knock-in mouse model, which are 140 repeats of CH triplet. And it's characterized as a slowly progressive model. We use 12-month-old mice, and we compare three groups: wild-type mice, heterozygous, and homozygous mice. So first of all, we characterize our model using histology. So we see accumulation of mutant huntingtin aggregates into HD mice in both heterozygous and homozygous mice. And we also show neuronal alteration into the striatum using spectroscopy. So you can see here metabolic profiles acquired in each mouse cohort. So we observe significant increase of glutamine associated with strong decrease of neuronal metabolites like Na and glutamate, meaning that in our animal, HD animal, there was probably neuronal suffering associated with astrocytic reaction. So as there was a decrease of glutamate, we try to investigate the interest of CEST imaging. So we acquire CEST images, and you can see here an example of CEST image, acquiring one mouse of each group at 3 different brain positions into the mice. So the most striking results, if you look on the bottom line, is the very strong decrease of CEST contrast in homozygous mouse compared to wild type. Interestingly, we see that for heterozygous mouse, the CEST contrast seems to be really in between wild type and homozygous mouse. However, here is just one example for one mouse of each group. So to have a better idea of what happened in the whole cohort, we calculated effect in each group. And as I mentioned before, the main advantage of CEST imaging is the very good spatial resolution. So to benefit from this advantage, we segmented the brain in 8 different regions of interest that you can see here, and we calculated the variation of CEST contrast between the wild type group and heterozygous mice, and also between wild type and homozygous. And we obtained this variation map of CEST contrast for each genotype. First of all, we can see a global decrease of CEST contrast in the whole brain, especially in homozygous group. Besides, we also see that the strongest decrease was measured into the corpus callosum, and it was the case in both groups. It is particularly interesting because we did not anticipate such variation in this brain structure. And also, it would have been impossible to measure it with spectroscopy, as I explained just before. To confirm these variations and to be sure it was not an artifact, we did the same calculation between 2 groups of wild-type animals and also with another mouse model of HD, which is R61. And this model is known to have preserved glutamate levels. And as you can see on the variation maps on the right, the strongest variation was measured to be only 4%, and it corresponds actually to the noise level. So this confirms that what we observe in KI-140 mice model was not an artifact, it was really due to alteration of these specific structures. So to conclude to this part, we demonstrated that MRS can provide a very rich and specific information about metabolic profiles, but it is limited by the voxel size and a priori positioning of the voxels. Interestingly, we were able to highlight alteration in unexpected brain regions, such as the corpus callosum. So it demonstrates that CEST can be very valuable in the context of HD, and it can, for example, give some clues to study the pathology in the clinical conditions in humans, because the techniques can be transferred to the clinic. Because obviously the following question is the technique transferable to clinics for patient investigation? I didn't mention it before, but CEST effect is field dependent. You can see here a linear relationship of CEST effect with the static magnetic field. Nonetheless, at 7 Tesla, the signal is still enough to detect glutamate with a good sensitivity. So it means that it is possible to perform CEST imaging on high field MRI scanner and clinical scanners, because 7 Tesla is now a clinical scanner, even if it's not very common in all hospitals. There are now good accessibility to 7 Tesla scanners. And it means that it's possible to do CEST imaging in human at 7 Tesla. And you can see here the first example of CEST map acquired in healthy volunteer. So you can see a nice contrast between white and gray matter into the brain of this of healthy volunteer. We also see a very low Greasiest contrast into ventricles because there is almost no glutamate into the CSF. So after this first proof of concept, people applied the Greasiest techniques to study different brain pathologies. And you can see here an example of the use of Greasiest to identify epileptic foci in non-lesional temporal lobe epilepsy. However, human Greasiest studies remain quite sparse, but I'm convinced that they are promised to be developed in the coming years. The last application of CEST imaging that I would like to speak about is CEST imaging of glucose. It's also called glucosest. As you may know, glucose is the main source of energy production, and the brain is the organ consuming the most of glucose. Numerous studies have already demonstrated that glucose metabolism can be altered in the context of neurodegenerative disorders. So the gold standard to study glucose utilization in vivo is to use PET. So it's positron emission tomography. So in PET, we use a radioactive analog of glucose, which is called FDG, where we have replaced hydroxyl group by a fluorine 18, which is a radioactive fluorine. So we inject this radioactive compound, which is quite expensive to the subject, so it can be a rat or also a patient. And we acquired this kind of glucose utilization map in vivo. The idea here would be to use just normal glucose, which is very cheap, and to use CEST MRI to monitor how glucose enter into the brain cells, and to how it's consumed by cells, in order to create cheap and radioactive free map of cerebral glucose metabolism. So you can see here an example of dynamic glucosest images acquired during and after perfusion of glucose solution in wild-type rats. You can see here that just after injection, glucosest signal increased due to glucose uptake by brain cells. And after perfusion, the signal decreased, reflecting either the consumption of glucose or the washout of glucose by cells. Interestingly, thanks to the very good spatial resolution of CEST, it is also possible to do the same in the mouse brain, which is impossible to do it with PET due to the limited resolution of the technique. You can see here a clear difference of glucose uptake in different cell layers of the mouse hippocampus. And interestingly, these layers contain very rich neurons, especially pyramidal neurons, which are glutamatergic neurons. For example, if you compare the glucosest image with the glucosest image in the same region of the brain, we see that region containing higher glucosest contrast, so it means that there are more neurons, they correspond to regions with higher glucosest uptakes, meaning that these regions are probably more active. And as we know that hippocampus is particularly affected in AD, glucosest could be a very good tool to monitor alteration in very small and specific region of the hippocampus in Alzheimer's disease. So in conclusion, I hope that I managed to convince you that CEST can be a very useful technique. And for me, CEST is a kind of toolbox to explore new contrast in MRI. It can be very powerful if we combine CEST and the good spatial resolution of CEST imaging with the high biological information provided by spectroscopy. So to me, CEST has a lot of potential applications, especially in the context of neurodegenerative disorders, and it's a very translational imaging modality. So now it's really the time to transfer with the technique from preclinical to clinical study. So I would like to thank you for your attention, and now the session is open to questions. Thank you again.
Thank you, Julien, for your very nice presentation and clear examples. We have now some time reserved for questions, as Julien already mentioned, and the first question is for Martin. Martin, could you describe how the imaging services of EATRIS work?
Yeah, thank you for the interesting question, Roseanne. I'm happy to explain how the infrastructure can be accessed to any type of user, whether it's a researcher at a non-profit institution or a company. We do take in service requests or research requests where we have a careful look together with any user at the project goals. What we will also do is do a translational assessment. So the scope of the work is clearly focused on the clinical translation. So if it is basic research, then it will not be within the scope of our activities. In the meantime, we're happy to have France joining in as well. And if we look to the process, the second step is a matchmaking step where we will take the request. So if you would have become interested by the versatile imaging just presented by Julien, we will sit together with our French partners, which will be easy, but we have more imaging sites and we will find the eligible sites. In the end, the client is in the driver's seat and will select the partner. And in the third step, we will in more detail discuss and explore the project and design the project. In the end, this can result in a project in a bilateral agreement between one of our institutions and yourself. And it could also be a multilateral contract. In the slide, there are a number of logos of organizations we work with to illustrate also the diversity of the clients we have. And there are several types of contracts that can come forth from such a collaboration, being it a service request or a multilateral, more complex overall framework agreement. I hope this explains a little bit how we work, and I hand over back to Roseanne to address the scientific question.
Thank you, Martin, for that. I have now a few questions for Julien. The first one is, what is the specificity of the glucose CEST measurements regarding glutamate, and how can we be sure that we are looking only at glutamate?
Yeah, it's usually a question that comes very often because all these molecules, all like glucose, lactate, and glutamate are all in the brain. So when you do CEST, it can be a mix-up of all these effects. But as I showed, you can select and set up your saturation module to selectively excite one exchanging group. So for glutamate, when you optimize the saturation pulse, we consider that 75% of the contrast comes from the glutamate. So the image that we obtain is not a pure glutamate image, but it's more true to say that we have a glutamate weighted image. But anyway, it's a good representation of glutamate variations.
Thank you for that. There's another question on how well do glucose and glutamate satisfy criteria for an ideal biomarker?
Yeah, it's a good question. So you have a good sensitivity and good quite good specificity. then it's quite robust because you can acquire your image in few minutes. Also, it's translational in the condition, for example, for glutamate that you have at least a 7 Tesla magnet, and it's absolutely non-invasive. So you can repeat the experiment over time. That there is no danger for the animal, especially for glutamate, because you don't have to inject anything. So I think for GluCEST, it's a pretty good biomarker, and now it's really the time for us to really evaluate the full potential of the techniques in different animal models, and then to have opportunity to do that in humans.
Thank you for your answer. There is another question about whether CEST MRI has been translated to human studies and whether there are any examples.
Yeah, so I've shown one example in the presentation in the context of epilepsy, and there is at least one other study performed at 7 Tesla again to study schizophrenia. So all these studies were performed in Philadelphia with the group which proposed first this method. Personally, here we don't have a clinical scanner, so I didn't have opportunity to evaluate the method in clinics. But in the literature, we have some example and which show that it's feasible, and it's really interesting because you can, in the context, for example, of epilepsy, you can really identify very small structure, and you can really look at the foci of the epilepsy.
Thank you for that. And there's one last question about whether you could elaborate a bit more on the nature and the range of CEST agents and their associated limitations.
So here is just an example concerning 5 different molecules, but actually you can image very different other different molecules. So this concern, what I show here concern endogenous metabolites. But there are some contrast agents also which provide CEST effect. But up to now, the size of these molecules are pretty large, so the application is quite limited for the study of brain pathologies. But with these molecules, you can really image very different biological processes. So, for example, as I showed with glutamate, which is mostly in the neuronal compartment, you can have access or study the state of neurons. With creatine, for example, you can measure the energy metabolism. With myoinositol, which is more related to astrocytic compartment, you can have an idea of what happened in astrocytes, for example. So there is really a very large panel of applications, and it's nice because except for glucose, you don't have to inject anything because it's already present in vivo.
Okay, thank you very much, Julien. We have now come to the end of this webinar, and I would like to thank you all for attending today's webinar, with a special thanks to our lecturers for their excellent presentations. Should you have any further questions, please do not hesitate to contact us via email. I would also like to point out that we have 2 more webinars in the upcoming 3 weeks. The next one is on November 20th and will be about microscopy for neuroscience, addressing how to choose the most suitable type of microscope for your research in the field of neuroscience. We hope to all see you again in 2 weeks, and thank you again. Have a good day.